Welcome, prospective students. My name is Christian Metallo. I've been a PI here in the Department of Bioengineering for almost 10 years. My background is in chemical engineering, and my laboratory studies metabolism and how it contributes and drives disease. We do this by studying metabolic pathways and tracing molecules through space, through biological systems and time. We do this in different disease models to try to identify potential therapeutic options. The approach we use is to quantify metabolic fluxes. And we do this by applying stable isotope tracers. These are non-radioactive molecules that are just a little bit heavier than the normal molecule, but otherwise are treated the same. We apply this to a number of different biological systems, ranging from cancer cells to stem cells and their derivatives, animal models, and in some cases it's humans through clinical collaborators. We apply mass spectrometry-based metabolomics to quantify isotope enrichment throughout the metabolome, and some simplified models to try to interpret these results and resolve how metabolism is functioning in a given disease state or system. We have a number of different areas of interest. The things I'll touch on today relate to cancer and neuropathy and macular disease. But if there's a core scientific theme that's emerging from my laboratory, it's that alterations in metabolic flux and enzyme promiscuity alter the lipidome to drive disease-related processes. Where does the engineering come into this? This is the same approaches that we use to try to quantify flows and residence times through continuous stirred tank reactors. But in our case, instead of biochemical reactors, we're looking at metabolic pools. In terms of applications, one area of particular interest is cancer. We and others have observed over time that two amino acids, serine and glycine, are critically important for tumor growth. There's evidence at the genomic level in certain cancers, at the transcriptomic level. This has been reduced to practice by us and others in that if you restrict serine and glycine from the diet, it will slow tumor growth dramatically. More recently, we've identified potential roles of lipid alterations in identifying these deoxydihydroceramide species by mass spectrometry that are elevated in the context of serine and glycine-free diets and accumulate within tumors to slow tumor progression. The overarching goal is to use our cell and animal models and mass spectrometry-based metabolomics to understand what are the mechanisms that drive this dependence on serine and glycine, and can we use this to dive deeper and identify new therapeutic options. At the same time, we've uncovered the fact that a prolonged treatment of a serine and glycine-free diet also drives peripheral neuropathy and retinal defects. And this occurs in mice. And as you can see, after 10 months on the diet, long-term treatment of the diet, the mice lose their ability to sense temperature and exhibit retinal defects as well. In this case, this is exciting because this mimics the phenotype in patients with very particular genetic alterations and have familial diseases of peripheral neuropathy and macular defects. But in this case, we're driving the phenotypes through the environment. And this kind of brings the overarching goal is to understand how different environmental factors and metabolic pathways and associated genes and transcripts converge at the systems level to drive disease. So I'm always looking to pick up excited grad students who are interested in learning techniques ranging from stem cell culture to mass spec-based metabolomics. So please shoot me an email if you're interested. Thanks.
Hi, my name is Carlos Rodriguez. I'm a second year in the Metallo Lab.
Hi, my name is Jeannie Gunga-Thurin. I'm a third year PhD student in the Metallo Lab. Welcome to the lab, and we're going to introduce you to tissue culture first.
This is our lab mate, Avi.
Hi. I'm Avi, I'm a fifth-year grad student here, and right now I'm just looking at some cells, putting some cells that are genetically engineered to knock out enzymes involved in acetyl-CoA synthesis.
Are you using anywhere to track how carbons are flowing, Avi?
Oh yeah, stable isotope tracers.
Best way to do
stuff.
it.
After using a metabolic stable isotope tracer to see flow through metabolic pathways, we can extract our cells and then analyze cells on the 3 GCMSs we have here to look at, for example, amino acids, the TCA cycle, and fatty acids, or cholesterol. And we've also developed methods for various other metabolites on these GCMSs. And using the stable isotope metabolic tracer, we can see where that isotope flows in different pathways and see isotope enrichment in different metabolites.
All right, sometimes when we're doing tissue culture, we're interested in what happens when the oxygen concentration is below atmospheric, or in areas where tissue or tumors, for example, might be hypoxic. And so we use the hypoxic chamber, where we can very accurately control the concentration of CO2 and oxygen so that we can recreate environments regarding oxygen concentration.
So we are now in the basement where we have our 2 in-house LCMSs. We have a triple quad LCMS and a high resolution Q-exactive LCMS. And these are used mostly to see more complex molecules like complex lipids such as sphingolipids and phospholipids, and even nucleotides and other sugars. We also have another lab member here.
Hi, I'm Grace. I'm a second year postdoc in the Metallo Lab, and I mainly use this machine, Q-exactive, with stable isotope tracers to look at cancer metabolism.
Hi everyone, thanks again for joining us on that brief Metallo Lab tour. That's just a brief view of what we do in the lab. There's a lot more to the world of metabolism and what we do in general. So if you have any questions or are interested, feel free to reach out to us or Christian, and welcome to UCSD.